And today we have our speaker coming from Minnesota, and we're very, very happy to have you, John, here. And I would like to ask maybe Sasha, you will do a formal introduction.
So, good morning, everyone. My name is Sasha Kolesnikov. I'm a scientist in Dr. Vladimir Kefelov lab here at UCI. And today it's my honor to introduce Dr. John Hulleman. Dr. Hulleman received his PhD from the Department of Medicinal Chemistry and Molecular Pharmacology at Purdue University in Indiana in 2008, where he studied the role of multifunctional protein deglycase DJ1 in Parkinson's disease. He then completed his postdoctoral training in the departments of chemistry and molecular and experimental medicine at the Scripps Research Institute in California in 2014. There, he investigated mechanisms by which the mutant form of glycoprotein called fibrillin 3 causes Doyne honeycomb retinal dystrophy, which is a disease phenotypically similar to age-related macular degeneration. And soon after that, he joined the faculty as an assistant professor in the departments of ophthalmology and pharmacology at the University of Texas Southwestern Medical Center in Dallas, where he developed his pretty broad research program. Since 2023, he holds a position of associate professor with tenure in the Department of Ophthalmology and Visual Neurosciences at the University of Minnesota in Minneapolis, where he is also a Larson Endowit Chair for Macular Degeneration. There, he also serves as a co-director of a high-throughput screening core at the Institute of Therapeutics Discovery and Development. The main goal of research in Dr. Hulleman's laboratory is to identify and pursue challenging and poorly characterized biological problems with broadly translatable implications for the treatment of ocular protein misfolding diseases. And more specifically, his lab aims to develop an understanding of the fundamental cellular and molecular mechanisms which underlie age-related and inherited retinal diseases, with an emphasis on disorders caused by secreted and extracellular matrix proteins, such as AMD and rare retinal dystrophies. To accomplish that goal, he applies his expertise in chemical biology, biochemistry, and various drug discovery approaches to restore compromised protein homeostasis associated with those pathologies. Dr. Hulleman's research is widely recognized in the field and is well funded by the NIH and many foundations, including Tom Foundation. He has published over 50 papers in reputable journals such as Journal of Neuroscience, PNAS, Journal of Clinical Investigation, Insight, Biochemistry, among many others. He has presented his findings as an invited speaker at many national and international meetings, perhaps most recent one being an International Congress of Eye Research in Buenos Aires, Argentina, where I personally had a chance to hear his interesting talk on how the inhibition of glycogen synthase kinase 3 protects against AMD-like retinal dystrophy. So with that, for our CTVR's distinguished speaker series, please welcome Dr. John Hulleman. And the title of his presentation today is Small Molecule Remodeling of the Extracellular Matrix Prevents AMD-like Pathology.
All right, well, thank you, Sasha, for that very nice and thorough introduction, as well as thank you to Grazina for the invitation to speak here today. So it's great to be back in Southern California. I was mentioning to people it was -13 with a wind chill in Minnesota on Wednesday. So I'll go anywhere where it's above zero. My lab at the University of Minnesota develops new treatments for inherited and idiopathic retinal diseases, including age-related macular degeneration and rare retinal dystrophies. And like Sasha said, we do so by leveraging our strengths in drug discovery, chemical biology, biochemistry, as well as gene therapy, ultimately to restore ocular protein homeostasis for disease intervention. And today I'll be specifically talking about a project in the lab where we used high throughput screening to identify a small molecule that is able to remodel the extracellular matrix and prevent AMD-like pathology in a mouse. So if we take a look at the eye, of course, there's a number of different tissues that must coordinate with each other so that we can sense light and see appropriately. There's the trabecular meshwork that helps regulate intraocular pressure, the lens, as well as the cornea, which provide the focusing power to focus light onto the retina, which is responsible for sensing light and transducing that light into electrical chemical potential, ultimately allowing us to see. And so my lab is specifically focused on the center portion of the retina called the macula, which is responsible for discerning fine detail as well as central vision, such as when we read, recognize faces, or even make sense of this presentation today. Now, of course, with ophthalmology, we have lots of cool tools and techniques that we can use to gain a structural and functional information about the eye in a non-invasive manner. And the retina is no exemption to this. We can perform simple fundus imaging or OCT imaging of the retina to provide a very detailed and informative picture. It looks very simple, but we can actually tell based on this picture the health or potential disease status of an individual. So if we take a closer look at the retina, we can see this comprised of a number of different cell layers, each of which are highly important and highly specialized. But arguably the most important, or at least the most studied layers within the retina include the photoreceptors, which are of course the light sensing portion of the eye, the retinal pigmented epithelium, or RPE, which are a critical layer that provide nutrients and energy sources to the photoreceptors, as well as degrade rod outer segments on a daily basis, and also participate in the visual cycle regeneration. Of course, beneath the RPE is the Bruch's membrane, a pentalaminar structure made up of collagen and elastin, and below that is the choroid vasculature. So my lab's focus is primarily within the RPE and Bruch's membrane interface. And the reason why we're interested in that is because that is the primary site of pathology for forms of AMD. Now, what does AMD really look like? Well, we can actually use this fundus images to actually determine the stage of AMD of an individual. AMD is characterized by the accumulation of drusen, these yellowish deposits underneath the RPE within the intercollagenous layer of Bruch's membrane, as shown here. And these drusen deposits are comprised of proteins, lipids, as well as trace minerals. Now, at this stage of AMD, this is pretty early on. There's very little visual impairment, but this disease can actually progress and the drusen can get larger and more disruptive, causing a higher likelihood of significant visual impairment. And of course, the disease can progress to even more advanced stages, such as geographic atrophy or wet AMD, which can cause rapid and irreversible blindness. But I'm not going to be talking about that today. My primary focus will be on identifying ways to prevent drusen formation, or at least the early stages of drusen formation. And we think that by doing this, we can actually potentially halt disease progression at an early stage where disease is not significantly affecting vision. And this is, of course, an important problem. About 200 million people worldwide have some form of AMD, and over about 90% have early to intermediate AMD, so about 180 million people worldwide. So what really causes AMD and what factors influence it? Of course, as any age-related disease, age is the #1, as of yet, unmodifiable risk factor for developing AMD. Of course, many of our choices, such as cigarette smoking, dietary choices, such as this high-fat, high-cholesterol, rich bacon, onion ring cheeseburger, can also affect AMD progression. And more recently, a study has demonstrated that alcohol consumption might actually affect GA progression. Low levels of moderate consumption of alcohol in males seems to protect against geographic atrophy, whereas high levels of alcohol consumption can actually accelerate it. And of course, there's always going to be a genetic component of these diseases. So 90% of AMD patients are Caucasian. And of course, GWAS studies have identified a series of genes with SNPs that can influence AMD progression and onset. So but what I'd like you to take from this slide is that AMD, of course, is very complex. multifactorial, and therefore pretty difficult to study within the laboratory within a reasonable amount of time. So to get around some of these issues, my lab has focused on what we call an early onset form of AMD. This is a disease that's caused by a single mutation in a protein called fibrillin-3, also known as EF-EMP1. And for the purposes of this talk today, I'll be just referring to it as F3. And individuals with this particular mutation can actually get very severe disease, very similar to AMD. Now, I will mention that this individual is a very rare case. This individual actually has 2 copies of this mutation and actually developed significant disease by 12 years of age. But most patients with this mutation actually get vision defects around 30 to 40 years of age. But nonetheless, we use this model as a way to study AMD within the laboratory within a reasonable time, because we can use it as a way to engineer cells as well as animal models that develop early features of AMD. And if we can do that, then we can potentially identify small molecules that might be able to prevent that pathology and determine whether or not they could be protective in the long run. So how does this particular mutation cause this early onset AMD? Well, we don't quite know, but we do know that there are a few indications that this mutation causes that might influence AMD progression, including. So we know that simple introduction of this mutation, or actually any other aromatic or disruptive mutation at that 345 residue, could cause significant secretion defects in simple transfection cell culture reagents or cell culture studies. Also, researchers from Eric Pierce's group have shown that essentially this protein, once it gets outside the cell, can form extracellular aggregates. So definitely it drives aggregation. And these aggregates may in turn trigger a complement activation through abnormal formation of the extracellular matrix. Additionally, we have recently showed that this mutation can also induce higher levels of NLRP3 activation, as well as production of pro-inflammatory cytokines, which I'll get into a little bit later in the talk. And Steven Seng's group has identified potential other mechanisms by which this mutation may influence AMD, including essentially inhibition of cholesterol efflux from cells, resulting in accumulation of neutral lipids within the RPE. So really, the R345W mutation may be causing disease through a number of different mechanisms, or a combination of all these. So luckily, back in 2007, Eric Pierce, as well as Lee-Hua Marmestein, independently generated 2 mouse models where they introduced either 1 or 2 copies of this R345W mutation. And in doing so, they observed that with essentially gene dosage, as well as with age, you start to get the accumulation of these basal laminar deposits, which are essentially harbingers of RPE stress and, for the lack of a better term, early stages of drusen formation. Of course, mice don't form drusen, but these nonetheless show an inclination for potential drusen formation eventually. For those of you who don't really look at EM images, I've given a nice little cartoon, a schematic to show where we're looking at, and here's Bruch's membrane shown here, the basal infoldings right on top of that, the RPE cells here, and then of course the photoreceptors on top of that. So it's very clear that this mutation is indeed pathogenic. Introducing it into mice does cause phenotypes, and other groups have also introduced this mutation into cell culture, such as Ruchir Singh's group, and have shown that indeed we can actually form deposits also in RPE cultures. But I'll be focusing primarily in validation of potential small molecules in the mouse model. So since this early onset AMD is an autosomal dominant disease, we speculate that we could actually lower the production of fibrillin-3, and that by lowering the production of fibrillin-3, we might be able to delay disease progression. Before we did that, we had to verify and convince ourselves that removal of fibrillin-3 itself had no detrimental effect on retinal physiology. So luckily, again, back in 2007, Lee-Hua Marmestein's group not only made a knock-in mouse model of fibrillin-3, but also did a knockout mouse. And this knockout mouse systemically started to show pelvic organ problems with increased inguinal hernias, shown here. But we did a more thorough characterization of the eye in these mice up to 15 months. And what we found was that essentially removal of fibulin 3 from these mice is extremely well tolerated, both as indicated by OCT, as shown here, by looking at flat amounts of RPE shown here, as well as a thorough characterization of retinal ganglion cell density, as well as optic nerve and ERG response. So we were pretty confident that removal of fibulin 3, or at least lowering the production of fibulin 3 in the eye, would be very well tolerated. And this was reaffirmed when we were able to obtain some data from one of 4 known individuals worldwide who have no fibulin 3. So this individual has a compound heterozygous mutation in fibulin 3 that actually causes them to have no detectable fibulin 3. As you can see, this individual has a bit of a different stature. They have more of a Marfan-like stature, a long, elongated face, hypermobile joints, and they also are prone to hernias, just like we observe in the mouse. Luckily, we were able to obtain some ocular imaging from this individual. This was during the COVID pandemic, so we were happy to get any sort of information on this individual. So we see that the fundus images look reasonably normal, as well as the OCT thickness looks reasonably good too, and corroborating our findings in mice that removal of fibulin 3 seems to be well tolerated, at least within the retina. So this drove us to then identify potential ways to reduce fibulin 3 in an attempt to prevent disease. And the way that we did that is by modifying fibulin 3 with a high-bit tag. And this high-bit editing allows us to follow endogenously produced fibulin 3. So it doesn't require any sort of overexpression analyses, and we think is more physiologically relevant and probably more translatable. So the origins of high-bit come from nanoluciferase, or NanoLuc, a small luciferase developed by Promega. It's about 100 times brighter than Firefly or Ranilla, and about 20 kilodaltons. So in and of itself, it's a really nice reporter enzyme, very bright, very stable. But Promega actually took this a step farther and actually separated NanoLuc into 2 different species, a high bit and a large bit. And independently, they are not luminescent themselves. But of course, when they are present together, they can actually reconstitute and actually form nanobit, which is luminescent. So you may be wondering, well, why would we want to do this? Well, the advantage of doing something like this is that this small portion here, this high bit portion, can actually be knocked into virtually any gene of interest. In our case, fibulin 3. And essentially serves as a nice handle to be able to follow and monitor the production of fibulin 3 very, very easily in a very simple plate-based assay. And this can be accomplished by supplying large bit in trans in the assay itself, or even producing large bit within the cells that also produce your high bit tagged protein of interest. And so we're pretty high on this high bit tagging. And I just wanted to mention that although we use this for high throughput screening, the uses of this high bit tag is actually quite wide. And you can use it for things such as protein of interest trafficking, stability determinations, and protein-protein interactions if you use small bit as opposed to high bit. So it's very versatile. We went ahead and did this. So we designed a strategy to knock in this high bit tag into fibulin 3, and that's shown here. Initially, we also included this 2x flag tag because we weren't quite sure whether or not this high bit tag was as good as people had said it was. So we added another epitope tag just in case. We performed editing in ARP19 cells, which of course we acknowledge aren't particularly good RPE cells, but nonetheless a good biological model that produce fibulin 3 and can be used for high throughput screening. Our editing efficiency was not particularly great, but reasonable enough, about 15% editing. And here's some genomic DNA that's been amplified. And the band here corresponds to the high-bit insertion that we verify by Sanger sequencing. And even the small amounts of editing can allow us to easily detect fibrillin-3 using high-bit blotting or even the high-bit assay. We performed a series of different validation steps, including siRNA and Sanger sequencing, to confirm the specificity of our editing. And you can see here with siRNA essentially knocking down the fibrillin-3 transcript, essentially reduces the high-bit signal down to baseline, indicating that we did indeed specifically edit the fibrillin-3 locus. So we're pretty happy with the performance of the assay so far. So we decided to miniaturize this assay into 3D4-well format. And we decided to begin screening for small molecules that might be able to reduce the production of fibrillin-3. So we initially started out with screening about a 2,000 molecule library, and that came from the Prestwick library as well as the NIH clinical library. And the idea behind using these libraries was to be able to identify a small molecule that we might be able to repurpose and maybe use more quickly or more effectively in patients. And some of these small molecules also have somewhat of a known mechanism of action to give us an idea of how this molecule was reducing fibrillin-3 production potentially. So here's an example of a 3D4-well plate. So each one of these wells is a different small molecule treatment. And you can see here that most of the small molecules have no effect on fibrillin-3, but there are some potential reducers as shown here. And of course, we always have to have a positive control for Feldman A. Long story short, we identified about 17 different small molecules that were able to significantly reduce fibrillin-3 production and validated 7 of those molecules in a 12-point dose response curve shown here. And 2 of these molecules, the AZD molecule as well as the CHIR molecule, were both GSK3 or glycogen synthase kinase 3 inhibitors. And since this was an enrichment in these apparent hits, we decided to continue on and follow up on the potential involvement of GSK3 in regulating fibrillin-3 production. So we went through the literature and randomly identified small molecules that had been characterized previously as GSK3 inhibitors and asked whether or not they actually had a similar effect on fibrillin-3. And indeed they do. So this biocompound CHIR 99021, as well as the CHIR 98014, which was identified in the original screening, all have an ability to reduce fibrillin-3 secretion as well as intracellular levels. And of course, the lithium chloride is a canonical GSK3 inhibitor, and it actually is the only GSK3 inhibitor that is FDA approved for use in humans. So we were pretty happy by these results, and we decided to focus primarily on this CHIR 99021 compound, which I'll call CHIR from now on, because it had a nice dose response throughout the different concentrations used, both intracellular as well as secreted, whereas the CHIR 98014 had a bit of a rebound effect, as you can see here at high concentrations. So we thought we had a little bit more playroom with the CHIR 99021. We verified that this molecule was able to also reduce not only the high-bit assay as detected here in a plate-based format, but also at the protein level using high-bit blotting, which is akin to Western blotting. And we verified that CHIR seems to have an effect, a significant effect on fibrillin-3 transcript levels, as shown here in qPCR. And we know that this reduction in fibrillin-3 is not due to causing toxicity, for example. We performed a series of different toxicity assays, cell titer glow, LDH, resazurin, and none of them showed any toxicity. That's always a concern, though, with assays where you're looking for a reduction in particular proteins of interest, that it might just be killing the cells. But in our case, it's not. So we not only did high-bit editing of ARP19 cells, but then we also performed these editing strategies in primary human dermal fibroblasts as well as mouse cells. And we found that essentially treatment of all those cells with CHIR led to a significant reduction in fibrillin-3 secretion as well as intracellular levels. So this is not just an ARP19 phenomenon. It is actually more biologically more widespread. So with these small molecules, of course, there can be significant potential off-target effects. And so we further validated that GSK3 was important through using siRNA, which of course will be more specific. There are two isoforms of GSK3, alpha and beta. We see that siRNA of those individually will actually significantly reduce fibrillin-3, but the combination is even better. And these results essentially parallel what we see with the GSK3 inhibitors previously. And of course, we validated those findings using Western blotting and high-def blotting shown here, and then quantified here, again, showing a nice parallel to the assay. So what is GSK3? You may be asking yourself that, and what does it really do? Well, arguably, it's probably one of the most promiscuous kinases within the cell and has lots of different effects on proliferation, metabolism, et cetera. Of course, I don't want you to take too much from this image other than it's involved in many different pathways. And one of the most characterized pathways is its involvement in Wnt signaling. So essentially, Wnt signaling can be triggered either through addition of Wnt ligand or even neurin. Alternatively, inhibition of GSK3 will actually release beta-catenin and will produce Wnt signaling also. So we were concerned about this because we know that Wnt signaling can have potentially pleiotropic effects, such as epithelial to mesenchymal transition, vascularization, as well as cell fate decision changes. So we wanted to see whether or not we could actually use these GSK3 inhibitors to reduce fibrillin-3, but without triggering Wnt signaling to avoid these potential pleiotropic effects. In order to do that, we developed a reporter line where we have a constitutively expressed M-cherry and then a conditionally expressed GFP. And so GFP in this case is proportional essentially to Wnt signaling. We then treated these ARP19 cells with a small molecule for a week, and we can see here a clear threshold effect after about 2.5 micromolar, where we actually start triggering Wnt signaling after that concentration. So we decided that we could actually use concentrations about 1 micromolar to avoid any potential Wnt triggering, and then ask whether or not that actually was sufficient to reduce fibrillin-3 production. And indeed it is. So we can see here that 1 week of CHIR treatment at 1 micromolar reduces fibrillin-3 production by over half. And again, this is not toxic. And we even developed another more sensitive reporter line that essentially produced firefly if Wnt was produced, or sorry, Wnt was activated. And indeed, this lower concentration of CHIR for a longer period of time did not trigger Wnt signaling. So we're pretty happy with those results. We know that CHIR has an effect on fibrillin-3, but then we asked the question more broadly, what else is an effect within the RPE cells? And so we did RNA-seq after treating ARP19 cells for 1 week with the CHIR. And what we found was that a number of different ECM-related components, such as collagens, lysyl oxidases, as well as other proteins involved in potentially sub-RP deposit formation, such as melatonin, or even complement component C3, were all significantly downregulated. In addition to, of course, fibrillin-3, which is shown here as EF-EMP1. Now, concomitantly with that downregulation, we also see that there are increases in differentiation associated genes such as tyrosinase and tyrosinase-related protein, as well as SERPINF1, which is PEDF. So it seems like we're reducing and remodeling the ECM while also promoting RPE differentiation. And these results were also corroborated in the gene enrichment analysis, where you can see a number of different processes primarily associated with the ECM or collagen production, all being significantly enriched in our analysis. So with that, we decided to take the next step, which is to determine whether or not CHIR could actually even get into the eye. So we performed a series of pharmacokinetic experiments using an IP-administered CHIR, 25 mg per kg, and then sacrificed mice at these different time points and harvested their plasma, liver, or their retina. followed by a sensitive LC-MS assay that we developed. What we found is that indeed the CHR can actually get into the retina, and the levels equate to about 1.6 micromolar at the highest level, which is great because we wanted to aim for approximately 1 micromolar to avoid potential Wnt triggering, at least based on our cell culture studies. But nonetheless, we were happy to see that it gets into the eye and at levels that we think would be actually therapeutically potentially beneficial. So we decided to develop a dosing regimen. So we took the early onset AMD mice, which are the R345W mice, aged them out to 8 months, and then treated them essentially 5 times a week for 1 month to determine whether or not CHIR could actually have a protective effect on basal laminar deposit formation. Before we did that, we verified that CHIR had no effect on ERG readings, both A wave and B wave. And of course, retinal histology looked identical to vehicle-treated mice, indicating that CHIR has no adverse effects on the retina, at least for this duration. But really, the big question was whether or not CHIR could prevent the main pathology observed in these mice, which is basal laminar deposit formation. Indeed, we were very excited to find that it can. So here's a zoomed-in EM image of vehicle-treated R345W mice at 9 months. And you can see here the beginnings of these little basal laminar deposits, as indicated by the arrows, and a concomitant decrease in the basal infoldings, which we typically observe with increasing basal laminar deposit formation. Now, as you can see here in the CHIR-treated mouse, in some fields of view, we do indeed see very small deposits. But if we do see any deposits, they're significantly smaller. And we see a significant reduction in the number of fields of view that have any deposits whatsoever. Moreover, you can tell that these RPE cells are quite healthy because they have really nice basal infoldings. So we're extremely excited by these results. And if you look up maybe a little bit closer, you'll actually start to see these little dots here in the EM. And each of these little tiny dots is actually a mitochondria. And we found that mitochondria were actually apparently significantly increased in the CHIR-treated mice compared to vehicle-treated mice. So this compound could be acting through a number of different mechanisms, not only through fibrillin 3 reduction, ECM remodeling, but also potentially through metabolism changes in RPE. So to summarize that portion of the project, we really think that hybrid tagging is a great, useful way to follow endogenously produced proteins of interest. It's a great discovery technique. Low-level, long-term CHIR essentially can significantly remodel the ECM and reduce the ECM burden. And that administration of CHIR can actually prevent basal laminar deposit formation in this early onset AMD mouse. So in terms of current directions where we're going from here, so here are some natural history graphs of essentially basal laminar deposit formation in these early onset AMD mice. What's very interesting is that in these mice, there's a huge jump in terms of the basal laminar size between 8 months and 12 months. And this is exactly where we treated the mice at 8 months. And so we think that we were treating the mice at a very critical time in the genesis of these basal laminar deposits. And that's probably why we were able to prevent them from forming in the first place. But I think a more clinically relevant question is whether or not we can actually induce reversal of these basal laminar deposits. Instead of treating mice at 8 months, what happens when you start treating mice a little bit later on when these basal laminar deposits have formed fully in terms of size and are increasing in number? Can we actually achieve a reversal of basal laminar deposit formation? And another aspect that we are currently testing is whether or not treating at 8 months for 1 month with CHIR could serve as a way to protect mice in the future. Are we treating at a critical time point to protect mice later on in life? And so we're doing a 1 month treatment with a 3 month pulse to see if we can prevent further deposit formation later on. So in the next few slides, I just want to go over some of the other projects that we're focused on. Not only can we identify small molecules using a high throughput, unbiased mechanism, but we can also identify other ways to treat these mice to potentially reduce basal laminar deposit formation using rationale-specific approaches. So I mentioned previously that one of the characteristics of these knock-in mice was that they actually have indications of NLRP3 inflammasome activation, as indicated here, as well as production of pro-inflammatory cytokines. We further demonstrated that they actually have a higher likelihood of infiltration of these IVA1 positive, potentially microglia, and they have increased immunoreactivity for NLRP3. So we rationed that maybe NLRP3 would be a good target to genetically remove and ask whether or not removal of NLRP3 could also protect against basal laminar deposit formation. And indeed, it does quite robustly. So we generated an R345W knock-in as well as NLRP3 knockout mouse and found that indeed these mice are significantly protected against basal laminar deposit size as well as a number of fields of use of coverage. Not only that, if we knock out the downstream effector, caspase-1, that produces fully mature inflammatory cytokines, we also get a nice robust protection as shown there. So there are multiple ways that we could actually approach this potential disease and potentially develop new treatments for it. So as I mentioned a couple of times, we're really high on high-bit tagging, and I hope you can see that high-bit could be useful for proteins of interest that you may be interested in. We have also high-bit tagged a number of different AMD-associated risk genes, including CFH, HTRA1, C3, as well as ApoE. And I've done this pretty successfully in ARP19 cells, as shown with these strategies. And I'm just going to focus a little bit on our new project on HTRA1 high-bit, where we were able to identify new small molecules that can regulate HTRA1 abundance also. So Brandy Williamson and Greg Hageman had demonstrated in the PNAS paper that essentially individuals with a risk allele of HTRA1, ARMS2, had a lower amount of HTRA1 produced within the macula RPE. And so identifying small molecules that might be able to enhance HTRA1 expression or even stability could be potentially useful in some of these individuals or patients. So we developed that high-bit tagging approach for HTRA1, went through all the series of validation steps, including the siRNA, Sanger sequencing, high-bit blotting, et cetera, and then miniaturize it to a high-throughput screening assay where we can identify potential enhancers or reducers. And we were able to identify this molecule, which is a diazo-containing dye typically used for vascular imaging, at least in the past, as a hit compound. More recently, this has been shown to have anti-amyloid and cardioprotective effects. We can see here that HTRA1 is increased with administration of the small molecule Chicago Sky Blue or CSB. And one of the good things about making all these different high-bit tagged cell lines is that they can actually serve as a nice control for each other. And so we can actually treat, for example, CFH high-bit cells with this CSB and see that there's no effect whatsoever on secretion or intracellular levels to gauge a degree of specificity. Again, we verified these results in high-bit blotting, but interestingly, we found that the effects of this compound are actually post-transcriptional. So we can see here in qPCR, there's no difference in HTRA1 transcript levels. Furthermore, the small molecule CSB has no effect on HTRA1 activity, but it does promote its abundance, thereby promoting overall higher levels of HTRA1 activity. And we think that this is due to interactions at the trimer interface of HTRA1 and promoting its stability through those mechanisms. So with that, I'd like to thank everyone who has contributed to the work. Sophie and Antonio did a lot of the work on GSK3. Sophie's a PhD student at UT Southwestern. Antonio's a PhD student at Johns Hopkins, and Grayson's a PhD student at Baylor. Of course, I won't be anywhere without key members of our lab, including Steffi and Emily, and new members, Nathan, Pravalika, and Anna. And I'd like to thank the funding mechanisms, especially the Tome Foundation, for their generous support.
Thank you, John. We will go with questions from the room and from Zoom as well. And as we're organizing our thoughts and preparing for questioning John, let me start with GSK3 kinase in knockout is lethal in mice. And so I think it's through the apoptosis in liver, I think the liver gets degenerated very rapidly. So this is systemic treatment. Are you concerned at all? That's my first question. The second, the compounds that you showed us, you used them up to 20 micromolar. Is there an issue in the cell culture assay with solubility of those compounds?
Could you say that last part again?
Is there an issue, problem of solubility? Because it looks to me from the structure, they're not very soluble compounds.
Right, right. So I think at very high concentrations, compounds like those diazo dyes, CSB, as well as Congo red, can precipitate. We didn't notice any of that at these lower levels, but that is a potential concern at higher concentrations, 50 micromolar to 100 micromolar. In terms of the GSK3 a question that you had, I believe those are developmental concerns, right? And that's knockout of both GSK3 alpha and beta. I think that there are viable alpha and possibly beta, but not the combination of the two. So I think there are possibilities to maybe knock down these different isoforms and see if they have any effects on basal deposit formation. But to address whether or not we have concerns about lethality or anything like that, in adults that might, for example, receive this small molecule. GSK3 inhibitors have been used a lot in the 2000s, 2010s as potential therapies for Alzheimer's disease, for neurological diseases, especially because GSK3 has a known effect on tau phosphorylation. And there were some clinical trials that were performed phase 1 that demonstrated that they were indeed safe, but not efficacious. So I don't think that surprisingly, there's not a significant safety concern, in my opinion, because these have been used a lot in individuals, but they just haven't been effective in preventing disease. So they haven't advanced to further clinical trials.
And all of those drugs fail, most likely because they couldn't go to a higher concentration where it will show really a problem. And my last question, please forgive me. Of course, I got very excited about your presentation today. You showed the treatment of cells with your drug that changes the transcriptome. Did you do experiment where you, let's say, brominate those compounds or add some other modification that loses the potency, and whether that changes in transcriptome also was observed? This is critical control because you can take those cells and treat them with aspirin, and they will have 50, 60 genes that change expression. I don't know how meaningful those data are from that perspective.
Yeah, I mean, we have not done any analogs of the CHIR compound as a control. Those are good suggestions.
Okay, we have a few questions from the Zoom. And the first one is John Young Si Han. Can you please ask your question?
Oh, I didn't know I was going to be on camera. So, John, I just had a quick question about, well, I had two questions. One was, I think I missed it, but did you say you made a high bid tag for ApoE?
Yep.
Oh, okay. Well, then you'll be getting an email from me from later. And I was curious about like your findings about the mitochondria in your treated mice. When you did your comparison, did you compare it to like an age-matched wild-type mouse? Or was the increase in the mitochondria number just relative to your vehicle control mice?
Yeah, it was the vehicle control mice. We did not do quantification of age-matched wild type in that case.
Okay.
But yeah, the results were robust enough for us to believe in them, I think. About twofold increase.
Good. So then do you think it was just because you cleared your basal laminar deposits, then you just had more nutrient availability? So then that led to kind of a partial rescue of mitochondrial numbers, things along those lines.
Yeah, I guess, I mean, we would have to do the age match control like you suggested to figure that out. My feeling is that my feeling is that it's probably independent of getting rid of those basal laminar deposits and actually having a direct effect on mitochondrial biogenesis. I mean, we saw with some of the viability assays that we did, even in cell culture, a nice significant increase in ATP production as well as mitochondrial reduction potential in simple cell cultures with the CHR treatment. So we think it's probably independent of the basal laminar deposit rescue.
Okay,
It
thank you.
may have kind of an influence on, general RPE health, but what I'm saying is that reducing the basal laminar deposits did not necessarily result in an increase in mitochondria, is what we think.
All right, thank you.
Thank you.
And another question from the Zoom, that would be Alan.
Hi, I thought that was a lovely talk, and thank you so much. I was curious, am I correct that you're saying that F3 is accumulating both intra and extracellularly? And if that's the case, what controls the half-life of that F3? Is it ubiquitin dependent or autophagy dependent? And did you see an effect on either of those proteolytic processes from the CHIR, the 99021?
Yeah, that's a very good question. Did you have another question?
A separate question. I think I saw a lot of lipid deposits in that 9-month-old CHIR treated retina. So I was curious about that. But could you talk about the first one first?
Yeah.
The half-life of the F3 and what regulates it.
Yeah. So that's a very good question. And that's something that we don't know, but are currently exploring. What happens to fibrillin 3 once it gets outside the cell and how it's handled within the extracellular matrix is pretty much unknown, as is the quality control machinery that deal with potentially misfolded fibrillin 3 inside the cell. So those are things that I can't quite comment on, but are things that we're exploring for sure.
Thanks. And the other one is, do you see accumulation of lipid deposits? Those are EMs. I thought you had a lot of lipid deposits I was surprised about when you treated.
Yeah, I mean, I guess that would go along with what Stephen Sang has seen before about neutral lipid accumulation within IPS RPE that have the mutation. And I think at later stages, we probably see lots of vacuolization also in these mice. So yeah, I think for sure that is another potential phenotype.
Is it a concern?
Sure. I mean, yeah. I mean, I'm sure that we don't want to have these huge amounts of lipid droplets for sure within the RPE, and they may cause further degeneration later on. And these mice that we showed were pretty early, but later on ones look a lot worse.
All right, let's get back to the room and question from here.
Thanks for your talk, Dr. Hulleman. So I had a question about the basal laminar deposits in these mice. Did you look to see if CHIR alters the composition of these deposits at all? Like, are they just smaller, or are they maybe composed of different maybe proteins or lipids?
Yeah, so for sure they're smaller. They're about twofold smaller if they are present. We didn't look at the actual composition. I think that's a bit of a tricky experiment to do. All we can say is that, what we see by electron density seems similar. So it seems like they're probably similarly composed, but smaller and fewer. But we can't definitively say that without, maybe doing some sort of unbiased approach like mass spec or things like that. But I think it's a bit challenging, especially when, the deposits in the CHR treated mice are very small.
Hi, John. This is Gulab here.
Yep.
Hi. I'm glad to see your recent work and congratulations on the paper. I was curious if I'm understanding correctly, the mechanism of GSK inhibitor is to inhibit the gene expression of the this, or is it at the protein level? And a second part of that, have you investigated the GSK, this particular GSK inhibitor on other misfolded, genes, something like
Yeah, it does appear it's acting transcriptionally. So we saw significant reduction in fibrillin-3 transcript, which was then corresponded to a reduction in secretion as well as intracellular levels. So just overall reduction in the production of fibrillin-3.
So this gene inhibition is specifically targeted to this particular one?
No,
Like
it's
a global pattern of inhibition of transcription or something.
Yeah, it appears to be more broad, but it's definitely not specific for just fibrillin-3 because we see other ECM proteins, fibronectin, collagens also being significantly downregulated at the transcriptional level.
Hi, Dr. Hulleman. Thank you so much for this wonderful talk. So my question is about the basal laminar deposits as well. So because the F3 mutated patients usually will present with this marfanoid habitus, so they have this hypermobility, which makes them prone to injury, and also this mitral valve prolapse. Do you anticipate that with a treatment that is so frequent and is also systemically, or that you pursue any investigations regarding cardiac health, and also with being prone to injury if the wound healing characteristics are still maintained?
Yeah, very good questions. So, the level to which the GSK3 inhibitors can reduce fibrillin-3, we would think would not influence essentially pelvic organ support. Essentially, we're knocking it down by about half or so, whereas those individuals or mice that have no fibrillin-3 that show the hernias are complete knockouts. So heterozygous mice, while prone to like these hernias, actually don't have significant issues with herniation, for example. So we don't think that the small molecule will actually have that dramatic effect on pelvic organ support and things like that within the individual because of the level to which it works.
Hi, my name is Hyodantan, and my question is about the risk of cancer with CHIR treatment. You mentioned that upon inhibition of GSK3, there's elevated levels of beta-catenin, and you've shown multiple genes that are downregulated and upregulated, some of which are involved in differentiation. So I was curious to know whether there's a risk of activating oncogenes with the elevated level of beta-catenin.
Yeah. So I mean, I guess in an ideal world, we would find a therapeutic dose that would avoid, Wnt signaling and avoid those types of pathways. So we would actually use a lower concentration to avoid that. So because we showed that essentially we can find a concentration where we can reduce fibrillin-3 but not trigger Wnt signaling. So I mean, ideally, in an ideal world, we would identify a small molecule that could work only within the eye, right, and not systemically, and it would avoid many of those other potential effects, especially when dosing might be a little bit tenuous.
Right. So my question was because the dosage that you used was less than 1 micromolar, and you do still see aberrant expression of multiple genes. So I was wondering if there was that risk, and also what is the possibility of depleting fibrillin-3, and how does that compare to using the GSK3 pathway in terms of the phenotype?
I'm sorry, can you reword the last part?
Yes. So I'm wondering what the contrast is between depleting fibrillin-3, because that, as opposed to targeting the GSK3 pathway. So maybe using ASOs or
right
methods too.
Right. Yeah, for sure. That's a possibility. And the advantage, of course, of ASOs or siRNA would be the mode of delivery could be relegated to the eye, for example. So those are possibilities. it's too early to say which one might be better. If just targeting fibrillin-3 would be better than using something like a GSK3 inhibitor that might also have other effects that might be beneficial, we don't know.
Hi, Dr. Hulleman. It's Marco Vassetto from Philip Kaiser Lab, and we met at NICER. My question is about actually, in AMD, you have structural and also functional changes. So my question is, with your compound, did you see a functional rescue in your animal models? Was it like specifically ERG or things of this kind?
Right. Yeah. Good question. So these mice actually don't have any ERG defects. So you can take them out to 18 months and they have no ERG defects. They do have some molecular indications of inflammation and things like that, but Eric Pierce's group as well as us have not found any ERG functional effects in these mice.
Both scotopic and photopic?
It was we mainly looked at scotopic.
Yeah, thank you so much for your really insightful talk. At the beginning, you kind of contrasted the difference between Doyne's honeycomb macular dystrophy, this EFMP1 mutation with macular degeneration. Can you comment, or do you know what the degrees of activity of fibrillin-3 are in the various flavors of kind of bread and butter macular degeneration? Because that might implicate the molecule therapeutically.
Yeah. So for sure, there is support that would suggest that fibrillin-3 wild type, for example, or at least non-mutated, not the R35W, seems to play a role in AMD progression. So fibrillin-3 surrounds pathogenic drusen in AMD patients, but not in non-AMD controls. Duplication at the fibrillin-3 locus predisposes individuals for AMD risk. So fibrillin-3 burden seems to be correlated potentially with AMD risk also. Our feeling is that this is just kind of an exacerbated form of AMD. And so we're hoping that, of course, what we find here can be applied to more idiopathic forms like what you're alluding to.
Okay, we have one more question from the Zoom. Tony, are you ready to ask your question?
I'm not sure whether that is me or not, but since we are a translational vision research program, what are your plans to possibly carry this into human clinical trials?
Yeah, well, I mean, from our perspective, I think, we'd like to complete some of the studies that we've already have in process with potential reversal of basal laminar deposits, for example, and potentially the pulse treatment, and see how those treatments really affect basal laminar deposit formation before we really think about, anything, greater than a mouse. So we're a little bit removed for sure from initiating human clinical trials, but it's always kind of in the back of our mind. But I think we'd like to see a few more data points, supporting this small molecule and the importance of the GSK3 pathway before we would do something like that.
It's beautiful work.
Hi, it's very interesting and mind, I would say, exercise for thoughts. I have a question about actually the role of fibrillin, and because it really surprised me that removal doesn't do anything. So does it have any response in stress, role in its response to stress or particular situations? What actually, do you see any phenotype in any conditions in your mice?
We see any phenotype in any conditions.
With your mice with downregulated or knockout of fibrillin-3 in the eye?
We don't in the retina. In the cornea, we have seen some cornea opacification as well as vascularization. So that is a possibility. But in terms of the retina, it looks.
But did you try to stress it, like for example, laser-induced neovascularization or?
No. We just aged them out to 15 months, and tried to thoroughly characterize them as best we could. But for sure, I mean, maybe under certain stressful conditions, there might be, a difference between the wild type and the knockouts.
Right, thank you, John. We learned a lot and hopefully gave you some ideas or things where you would like to go. All right, let me join all of you to thank John.